Welcome to the deep dive. Today, we're analyzing, well, really the biggest medical story of the decade, GLP-1 based therapies.
Hmm.
If you follow the news at all, you already know the headline. These drugs, we're talking about semaglutide, tirzepatide, they have this almost unprecedented power
Yeah, it's quite remarkable.
for managing type 2 diabetes, obviously, but also for achieving massive weight loss. You often see numbers like, what, over 10% for semaglutide.
That's right. And climbing towards maybe even exceeding 20% for tirzepatide in people living with obesity. It's significant.
But our mission today really is to move past just the scale and the blood sugar numbers. We want to unpack the truly stunning underlying story here.
Which is,
These unexpected clinical benefits that seem to go far, far beyond just shedding pounds. We're looking at a stack of sources suggesting these aren't just metabolic drugs. They might be powerful, broad-spectrum anti-inflammatory agents.
and that's really fascinating because the central biological finding, the thread connecting all these unexpected benefits, seems to be inflammation. Our sources are pretty clear. Activating this GLP-1 receptor signaling, both acutely and chronically, it reduces systemic and tissue inflammation. We see this in human patients and in animal models.
And critically, it's not just about the weight loss causing less inflammation, right?
Exactly. Crucially, this anti-inflammatory action happens through mechanisms that do depend on weight loss, yes, but also through mechanisms that seem completely independent of weight loss.
Yeah.
And this intrinsic effect, this weight-independent part, is likely the engine driving this rapidly expanding clinical scope we're seeing.
Right, because they're already proven to reduce rates of major cardiovascular events.
Correct. And chronic kidney disease, too. These aren't just small effects either.
And the list of potential applications just keeps getting longer, it feels like.
Absolutely. I mean, they're under intense investigation right now for conditions like metabolic liver disease, and we'll probably call it MSH, for metabolic dysfunction-associated steatohepatitis. That's the newer term.
Okay, MASH, got it.
But also things like arthritis, substance use disorders, even neurodegenerative disorders like Parkinson's and Alzheimer's. The sheer breadth of the potential impact is, well, it's genuinely astounding.
Okay, let's definitely unpack this. Before we jump straight into the drugs themselves, maybe we should back up. Let's understand the body's natural system first. Where does this GLP-1 hormone even come from, and why is it linked to inflammation naturally?
That's a great place to start. It all begins in your gut, specifically with intestinal cells called L cells.
L cells, okay.
You can think of these L cells as biological sensors. They're constantly monitoring what's going on inside the gut. And a key function is acting as pathogen and inflammation sensors.
So they're like little guards.
Sort of, yeah. They don't just respond to nutrients and bile acids, the normal digestive signals. They also respond vigorously to signs of trouble.
Like what kind of trouble?
Well, things like microbial metabolites or even directly to pro-inflammatory molecules. We have very specific examples showing this direct link.
For?
Okay, so if you give mice or even observe in humans something like LPS, that's a bacterial endotoxin, a strong signal for the immune system.
That triggers inflammation.
Exactly.
Yeah.
Or if you administer pro-inflammatory cytokines directly, like IL-1 or IL-6, you see a rapid spike in circulating GLP-1 levels.
Rapid.
We're talking within about 3 hours. It's fast, both in animal models and observed in humans.
That sounds like a lightning-fast warning system. The body's using GLP-1 as an immediate defensive measure almost.
Precisely. And it fits with clinical observations, too. In humans, circulating GLP-1 levels actually correlate with the severity of critical illnesses.
Such as?
You see elevated levels in conditions like sepsis, end-stage kidney disease, acute respiratory failure. It strongly suggests the body uses this endogenous natural GLP-1 as an immediate, maybe protective, response to serious illness or inflammation. It's a reactive system.
Okay, that groundwork is essential. Because now we get to the core surprise of this deep dive, right? The fact that these drugs, these therapies, seem to have an anti-inflammatory effect that's somehow separate from the weight loss. And since we know weight loss itself is strongly anti-inflammatory, how do we actually prove, how can we be sure that these drug actions are truly weight loss independent? That seems key.
It is key. And we primarily track this using C-reactive protein, or CRP. It's a common, pretty reliable marker for systemic inflammation.
Right. Doctors measure it all the time.
Exactly. And we know that basically all the major GLP-1 medicines Exenatide, liraglutide, semaglutide, tirzepatide, they all reduce plasma CRP levels in people with type 2 diabetes or obesity.
Okay, but again, the question is why? How do we confidently rule out weight loss as the main reason for that CRP drop? This feels like the critical separation point.
It really is. And we have basically two main lines of evidence pointing to independence. First, the acute effects.
Meaning how quickly things happen.
Yes. A single dose of exenatide or semaglutide was shown to reduce circulating inflammatory markers, like TNF-α, in mice within hours.
Hours.
Hours. Far, far faster than any meaningful weight loss could possibly occur.
Okay, that's compelling.
And furthermore, in lab studies looking directly at human immune cells, PBMC's exenatide rapidly inhibited the binding of NF-κ to DNA.
And NF-κB is like a master switch for inflammation.
It's a crucial one, yes. It controls the expression of many inflammatory genes. So inhibiting its binding quickly points to a direct, rapid cellular mechanism, not just a consequence of slower metabolic changes.
So the sheer speed is strong evidence in animal models and cell cultures. But what about longer-term human data from the big clinical trials?
Right. The clinical data backs this up, too. If you look at the big semaglutide trials like SUSTAIN and PIONEER, they actually analyzed how much of the CRP reduction could be explained by the measured improvements in glucose control and the weight loss. And it turns out those factors could only explain, at best, maybe 20% to 60% of the CRP drop.
So a huge chunk, maybe 40% or more, was just unaccounted for.
Unaccounted for by those metabolic changes alone, suggesting something else is going on.
Okay, that's significant.
And maybe even more compelling was the PIONEER 2 trial. This one directly compared oral semaglutide against another type of diabetes drug, empagliflozin, an SGLT2 inhibitor.
Okay.
Semaglutide reduced CRP by about 30%. Empagliflozin, which, importantly, caused a similar amount of weight loss in that trial, around 4%.
Yeah.
It had virtually no effect on CRP levels.
Wow. Okay, same weight loss, totally different inflammation outcome.
Exactly. That really isolates the effect. The reduction seems intrinsic to activating the GLP-1 signaling pathway itself, not just a general effect of losing weight or improving blood sugar.
So if the magnitude of this benefit is largely unexplained by weight loss, I mean, how fundamentally does that change how we should think about these drugs?
It changes things significantly.
It means we really have to investigate these dedicated, rapid, anti-inflammatory mechanisms that must be at play. It's not just a bonus effect.
Precisely. And it leads us to ask, how does it work? Are there direct effects on immune cells? Or is it more of a central, indirect control, maybe governed by the nervous system? Or both?
Let's tackle the direct actions first. Does GLP-1 directly talk to immune cells?
Well, the picture is a bit complex. Generally speaking, GLP-1 receptor expression seems to be pretty low on most peripheral immune cells.
Oh, okay.
But it seems to be quite critical in specific subsets, particularly of certain types of T cells.
Which ones?
The data points strongly towards effects on gut intraepithelial lymphocytes, or IELs. These are specialized immune cells located right there in the gut lining, sort of on the front lines.
Makes sense given that GLK-1 comes from the gut L cells.
Exactly. And GLP-1 medicines appear to suppress specific T cell activity, especially within these IEL populations.
Now, I remember reading there's some technical difficulty here, something about detecting these receptors.
Yes, that's an important note of caution from our sources. It's actually technically quite difficult to reliably detect the GLP-1 receptor protein on T cells.
Why is that?
Apparently, many of the commercially available antibodies the researchers use don't work very well for this specific receptor on these specific cells.
So studies might miss it or get false results.
It means you need really rigorous scientific validation, like using control studies where the receptor is genetically knocked out, to be absolutely sure what you're seeing is real. It requires careful work.
Okay, assuming the receptor is there on some T cells, can you give us a mechanism nugget? How does activating it actually dampen that T cell's inflammatory response? What's it physically doing?
Yeah, the research suggests GLP-1R activation dampens what's called proximal T cell receptor signaling.
Meaning?
Think of it like this. When a T cell gets activated, there's a whole cascade of signals inside the cell, like dominoes falling. GLP-1 signaling seems to interfere right near the beginning of that cascade.
Okay.
Specifically, it inhibits the adding of phosphate groups, which acts like an on switch for key signaling molecules called ZAP70 and SLP76.
ZAP70 and SLP76. Got it.
By hitting those early switches, it effectively reduces the T cell's ability to produce major inflammatory cytokines downstream, things like interferon gamma, TNF-alpha, and IL-2.
So it's like putting a brake on the T cell's inflammatory engine right near the ignition.
That's a good way to put it, a powerful brake on localized inflammation.
Okay, so that covers direct local effects, but you mentioned maybe low overall expression. If the direct effects on most immune cells are limited but measurable, where's the main control center for the systemic anti-inflammatory action coming from?
This is where it gets arguably even more fascinating. It takes us to the central nervous system,
The brain.
the brain. There's strong evidence that GLP-1 medicines can acutely attenuate systemic inflammation, like the kind caused by that bacterial endotoxin, LPS, through central mechanism.
How do we know it's central?
Because these effects absolutely require GLP-1 receptor expression, specifically in brain neurons. If you knock out the receptor just in the neurons, that rapid anti-inflammatory effect is lost.
Wow. So the drug acts in the brain to essentially tell the entire body, calm down, stop being so inflamed. That's like the definition of neuroimmune crosstalk.
It is, exactly. The brain seems to be acting as a crucial control hub for this systemic anti-inflammatory signal generated by GLP-1 agonism.
Incredible.
And it's complex, too. This neuroimmune pathway seems to require intact signaling through other neurotransmitter systems, specifically alpha-1 adrenergic receptors and delta-opioid receptors seem to be involved in relaying that anti-inflammatory message from the brain.
So it's a sophisticated whole-body override switch triggered by GLP-1 in the brain.
That's what the evidence points towards, yes.
Eh.
A really elegant system.
That concept, a central anti-inflammatory override that helps explain why the clinical benefits seem so widespread, right, across totally different parts of the body.
Precisely.
So let's shift focus now and actually look at some of those wide-ranging clinical applications. Starting with the heart, the cardiovascular benefits, we know they're there.
Yes, in cardiovascular health, GLP-1 provides protection through multiple routes. Some is weight loss dependent, obviously, but there are also clear weight loss independent anti-atherogenic effects.
Meaning it directly fights plaque buildup.
Seems so. For instance, studies in mice prone to atherosclerosis showed that liraglutide reduced the size of arterial plaques. And importantly, this happened even when the control mice were carefully weight matched through diet restriction.
The weight matching again. Key control.
Very key. And also, the relatively rapid appearance of cardiovascular benefits in the large human outcome trials, often seen earlier than maximal weight loss, strongly suggests these faster-acting, potentially brain-mediated anti-inflammatory actions are central to the heart protection.
Okay, makes sense. Now, moving up from the heart to the brain itself, neuroinflammation is a huge issue, a hallmark of devastating diseases like Parkinson's and Alzheimer's. What have we learned preclinically there?
Preclinically, there was some exciting work. For example, a modified long-acting exenatide analog called NLY01. Right. It showed promise by reducing the secretion of pro-inflammatory cytokines from microglia. Those are the brain's specialized immune cells.
Okay.
And by doing that, it seemed to limit the conversion of nearby astrocytes, another brain cell type, into a harmful neurotoxic state called the A1 phenotype. This A1 state is linked to disease progression, particularly in models of Parkinson's disease.
So it calmed the brain's immune response and protected other cells. Sounds promising. But the clinical translation, that's been tougher, hasn't it?
It has, unfortunately. While some earlier, smaller trials with first-generation GLP-1 drugs like exenatide or lixisenatide did show some modest positive effects on Parkinson's motor scores.
Yeah.
A more recent, larger trial, specifically with that brain-penetrant drug, NLY01, unfortunately showed no significant clinical benefit in Parkinson's patients.
That's disappointing.
It is. And the results for Alzheimer's models in animals are also a bit mixed. So the underlying anti-inflammatory mechanism in the brain seems plausible, but actually getting the right drug to the right place at the right time to make a clinical difference, that remains the challenge there.
A common story in neuroscience, sadly. Okay, let's move down to the abdomen. The liver, specifically MASH, metabolic dysfunction-associated steatohepatitis. What's the news there?
The news there is actually very positive. In a phase 3 trial looking at semaglutide specifically for MASH,
Yes.
the results were quite striking. The drug achieved resolution of the steatohepatitis, the inflammation and liver cell damage without worsening of fibrosis in 63% of patients.
63% compared to placebo.
Compared to only 34% in the placebo group, that's a significant difference.
Wow.
And considering how few effective treatments we currently have for MASH, this is potentially a major breakthrough for liver disease.
Definitely sounds like it. Okay, how about the kidneys? The data for chronic kidney disease, CKD, has also seemed pretty powerful from the headlines.
Absolutely powerful. The source material actually connects the absence of GLP-1 receptor signaling in mice directly to accelerated kidney damage.
How?
It seems to be associated with increased levels of something called AGs, advanced glycation end products. These are basically sticky, damaged molecules that build up and harm tissues, including the kidneys. So the natural GLP-1 system seems protective.
I see. And clinically?
Clinically, the evidence is robust.
Yep.
The huge FLOW trial, specifically designed to look at kidney outcomes with semaglutide.
Right, I remember that one making news.
It demonstrated that once-weekly semaglutide reduced the risk of the primary composite outcome, which included CKD progression, kidney failure, or death from kidney or cardiovascular causes, by a very significant 24% over about 4 years.
24%. That's a major risk reduction for a really serious condition.
Huge. A very important finding for patients with diabetes and CKD.
Okay, finally, one more area that's maybe less expected. Joint health. We're seeing signals related to arthritis and osteoarthritis.
Yes, and this is where we see perhaps the clearest evidence of a local anti-inflammatory effect, separate from systemic changes or weight loss.
How so?
Well, preclinical models are very compelling here. One study showed that a single injection of liraglutide directly into the knee joint of mice with induced osteoarthritis.
Just one shot into the joint.
Exactly. It reduced their pain sensitivity and the synovitis, the inflammation of the joint lining. And crucially, this happened without any change in the animal's body weight.
Ah, so it must be acting right there, locally in the joint.
Precisely. It points strongly to a direct, local anti-inflammatory effect within the joint tissue itself, independent of weight.
And is there clinical data to support this in humans?
There is. The STEP 9 trial specifically looked at semaglutide in people who had both obesity and knee osteoarthritis.
Okay.
And it demonstrated a significantly greater reduction in pain scores compared to placebo. Using the WMAT index, a standard measure for osteoarthritis symptoms, the semaglutide group had a reduction of about 41.7 points, compared to only a 27.5-point reduction in the placebo group. So again, strong efficacy suggesting it's helping with the inflammation-driven pain in the joint.
Across the board, then heart, brain, liver, kidney, joints, this inflammation link seems real. Now, we have to talk about the newer agents, especially tirzepatide. It's a dual agonist, right? Hits both GLP-1 and GIP receptors.
Correct. GLP-1R and GIPR.
Does adding that GIP component change the anti-inflammatory profile? Does it add anything?
It seems like it might, yes. Tirzepatide appears to have distinct acute anti-inflammatory effects that could well be mediated by the GIP receptor activity.
How did I know that?
Well, remember that experiment where knocking out the GLP-1 receptor in brain neurons eliminated semaglutide's acute anti-inflammatory effect?
Yes.
They did a similar experiment with tirzepatide, and interestingly, even with a neuronal GLP-1 receptor knocked out, tirzepatide's acute anti-inflammatory effect persisted.
Ah, so the GIP receptor must be picking up the slack, providing that effect.
That's the strong implication. It suggests the GIPR component is contributing, possibly independently, to the overall systemic anti-inflammatory action of tirzepatide.
And does that fit with what we know about GIP biology generally?
It does, actually. Other studies, mostly in mice, have shown that losing GIP receptor signaling is associated with enhanced inflammation, for example, in fat tissue, and also with increased atherosclerosis. So it supports the idea that GIP signaling itself normally plays an anti-inflammatory role. Adding it back with tirzepatide might boost that effect.
Okay, so this really brings us back full circle to that fundamental question we set out with. Is metabolic control the weight loss, the better blood sugar? Is that alone sufficient to explain this incredibly broad efficacy across the heart, brain, liver, kidneys, joints?
And based on the evidence we've discussed, the answer seems to be a pretty definitive no.
No.
Clinical trials consistently show benefits,
Yeah.
particularly the anti-inflammatory marker reductions and sometimes even clinical outcomes, occurring even in people who don't lose substantial amounts of weight or faster than weight loss would explain.
Right.
This really underscores the necessity of digging deeper into these non-metabolic mechanisms, the direct immune effects, the vascular effects, the crucial neuroimmune crosstalk, to fully grasp why these therapies are working so well across so many different conditions. It's not just about metabolism.
Before we wrap up on this incredible spectrum of benefits, just for balance, we should probably quickly mention any associated inflammatory adverse events. Are there any downsides on the inflammation front?
That's a fair point. It's
Wow.
important context. While these drugs are overwhelmingly anti-inflammatory in their effect, One specific inflammatory adverse event that is associated with them is cholecystitis.
Inflammation of the gallbladder.
Correct. The risk is elevated, though still relatively low. However, it's also crucial to note that early concerns about a potential link to pancreatitis inflammation of the pancreas have generally not been substantiated in the large long-term clinical outcome trials. The data there is largely reassuring.
Okay, good to clarify. So what a deep dive this has been. The whole narrative seems to have fundamentally shifted, hasn't it, from just thinking about these as diabetes and obesity treatments?
Absolutely. I mean, the clinical expansion of these GLP-1 based medicines into areas like MSH, CKD, potentially neurodegeneration, maybe arthritis, organ protection.
Yeah.
It strongly suggests we are witnessing the emergence of a genuinely powerful, broad-spectrum anti-inflammatory therapy class.
Not just a side effect, but a core mechanism.
Potentially a core mechanism driving much of the benefit beyond pure metabolic control. It's really quite paradigm shifting.
It really is. Which leaves us with a final provocative question for you, the listener, to think about. We know that combining GLP-1 with other hormones like GIP and tirzepatide, but also potentially glucagon or amylin in drugs currently in development, modifies its effects.
The multi-agonists.
Right. So as we develop these even more complex multi-agonists, how will these combinations further refine or maybe even target these anti-inflammatory actions? Could we one day see targeted anti-inflammatory GLP-1 pharmacotherapies, therapies designed specifically to treat inflammatory conditions, maybe even ones completely separate from type 2 diabetes or obesity? Think about that potential.